mode.
Hello, welcome to Prairie Conservation Action Plan's Prairie's Got the Goods Week. My name is Caitlin Rose, and I am the Stewardship Coordinator with Saskatchewan Prairie Conservation Action Plan, or PCAP. Be sure to check out other events going on for Prairie's Got the Goods Week. Don't miss Sherry Grant's presentation tomorrow at noon about Canadian beef from a producer's perspective. Wednesday at 3 p.m., Paige Englot will be presenting about a loose Canada, working with farmers to sustain agriculture, wildlife, and natural spaces for all Canadians. Check out the PCAP website for more information about these events. I would like to take a moment to note that in-kind support for this project has been given by the University of Alberta. This project was undertaken with the financial support of the Government of Canada and the Federal Department of Environment and Climate Change Canada. Now, a bit about our presenter. Karen graduated from the University of Western Ontario with a Bachelor of Science in Environmental Sciences, where she completed an undergraduate thesis that initially interested her in researching plant-soil relationships. Karen completed her PhD at the University of Guelph, where she studied soil microbial communities with molecular methods under the supervision of Dr. Kari Dunfield in Ontario Agro-Ecosystems. Karen moved to Alberta in May to start her postdoc position under the supervision of Dr. Sylvie Quideau, Cameron Carlyle, and Edward Bork. She is currently working on two NSERC CRD-funded projects examining the recovery of mixed grass prairies under high-voltage power line construction. Specifically, Karen is studying the recovery and resilience of soil microbial communities after industrial disturbances related to power line construction, and how changes in these communities relate to mixed grass recovery under proposed mitigation practices. Before we begin, I would like to mention that if you have any questions during the presentation, please type it into the questions section of your webinar dashboard. Questions will be answered at the end of the presentation. I'd like to turn it over now to Dr. Karen Thompson.
Hi, Caitlin. Thank you very much. I will just pull this up. Okay. So, as Caitlin said, my name is Karen Thompson, and I'm happy to be here today to talk about a project that I'm working on through the University of Alberta. And I just wanted to kind of preempt this by saying I'm a soil microbial ecologist, but I'm also going to be presenting some plant community data that was collected by some great master's students we have out here who are also involved in these projects, Cassia James and Faiza Najafi. So on top of all of these profs working here, this is a lot of master's students as well. So we're going to start with a kind of background on our grasslands, which we heard a bit from Edward yesterday in his webinar. And so grasslands are found on approximately 43% of agricultural land in Alberta, and this makes their long-term conservation and sustainable management a priority. So native grasslands provide numerous ecosystem goods and services, including carbon storage, water purification and flood mitigation, biodiversity conservation, and the provision of wildlife habitat, as well as the provision of forage and livestock production. However, the remaining area of grasslands in Alberta ranges from 5 to 40% of its original area. So many of the goods and services provided by Albertan grasslands heavily rely on the healthy functioning of their prairie soils. And soils deliver and moderate ecosystem services through their healthy functioning. So the functions of a soil includes things like regulating water fluxes, moderating biogeochemical cycles, which provides nutrients for plants and also influences the Earth's climate, as well as providing habitat for numerous plants, animals, and microorganisms. So it is in our plants, or in our planet's and within our population's best interest to ensure that our soils are healthy so they can continue to provide these valuable services. So overall, a healthy soil will equal healthy ecosystems and therefore healthy grasslands. However, disturbances to native grasslands are steadily increasing, and this threatens the maintenance and conservation of our remaining grasslands. So disturbances to grasslands can include things like land use conversion to agricultural cropland, overgrazing, the use of these lands for recreation, as well as urban industrial development, which includes that associated with energy extraction, processing, and transmission. So across Alberta, urban regions are increasing in size and frequency, and this leads to sharp increases in energy demand. So as a result, infrastructure projects are continually being built within and throughout grasslands, and this can impact the long-term sustainability of these areas. We've seen in other systems, such as agricultural soils and forest ecosystems, that when large-scale disturbances occur, they often influence both the plant communities as well as the below-ground abundance and diversity of soil microbial communities. And this in turn can change many of the different ecological processes within these systems and the overall associated functioning of the ecosystem. So I'm specifically talking today about one project that's been conducted at the Mattheis Research Ranch, so I'll give you a bit of background on this. So from 2014 to 2015, ATCO constructed a transmission line across southeastern Alberta, and this provided a novel research opportunity to assess and refine best management practices, or BMPs, for transmission line construction and associated industrial disturbances. So the government of Alberta recommends the use of access mats, which are shown in this lovely picture taken by one of our other PhD candidates, Lysandra Pyle. So the government recommends using these access mats, which are shown here, to decrease the negative impacts of industrial disturbances on the prairies. So these access mats have been kind of one of these solutions that have been proposed to minimize these disturbances, and these mats can come in various material types as well as sizes. So we looked at one version of access mats that's made of 3 layers of spruce boards, and as I said, they can be interconnected, like in this picture here, to create roadways, and they were originally developed to provide access over soils within the boreal forest. So these mats weigh approximately 750 kilograms each and are roughly 2.5 by 4.5 meters in size. So the idea behind these mats is that they redistribute weight of heavy equipment traveling and working on them. They provide a durable surface to prevent soil eroding and soil compaction, as well as the ripping of vegetation. However, overall, these mats are a pretty huge expense for the companies using them. And depending on the scale of project, this can range up into the millions of dollars per year for transporting them to site, setting them up, and then taking them away from the site. So the government of Alberta has suggested guidelines for access mat use in order to mitigate the impacts of heavy traffic disturbance. However, despite all of this government support and use in industry overall in Alberta, there have only been 2 what we call gray literature reports, so not peer-reviewed, that document apparent benefits of access mats, and these studies were conducted in Wyoming. Regardless of this, the government of Alberta still supports the use of these access mats. So when ATCO created this transmission line from 2014 to 2015, they brought access mats in order to create durable access roads and construction platforms. However, after completion of this power line, when the mats were removed, soils were often bare with compacted soils, and marginal vegetation recovery was initially seen. So this gave us lots of questions about whether these were actually a proper mitigation practice. So some of the impacts seen due to these access mats at the Mattheis Research Ranch after access mat removal can be seen here in the bottom right picture. So these mats may physically crush vegetation and block sunlight, therefore preventing the plants from growing and photosynthesizing. And gas exchange and water infiltration is also likely reduced. So these impacts may in turn influence also the functioning of the soils underneath these mats, and therefore potentially influence the overall ecosystem goods and services provided by these grasslands. So for example, these mats may change nutrient cycling and therefore influence greenhouse gas emissions and soil carbon stabilization. So in general, the long-term impacts of using these are fairly unclear. However, they were initially proposed as this mitigation practice because previous studies, such as this one from Wilson in 1988, showed that traffic applied during the growing season of plants may produce more bare ground, which can lead to invasive species or non-native encroachment, as well as contributing to vegetation being crushed and potentially ripped out. So we basically wanted to be able to assess the impacts of both direct traffic on the prairies as well as access mat use paired with direct traffic on the mixed grass prairie, both plant and soil characteristics, including our microbial communities. We also wanted to evaluate the effects of the season of access mat placement as well as the duration of access mat placement on these different communities and characteristics. So in particular, we wanted to assess the effects of direct traffic on soil and plant characteristics, as well as assess the mitigative potential of access mats to decrease the negative impacts of traffic on mixed grass prairie. And we also wanted to try and establish appropriate guidelines for seasonal placement and duration of access mats if they do in fact alleviate the impacts of traffic. So the overall goal of our project was to try and understand the specific management practices that help mitigate impacts of grassland disturbance in order to promote grassland recovery after disturbance and therefore maximize the ecosystem goods and services provided by these agroecosystems in the long term. So the project I'm going to be speaking about was conducted at the Mattheis Research Ranch, which is located in southeastern Alberta, just about 230 kilometers north of the US-Canada border. And this is part of the University of Alberta and the Rangeland Research Institute. So when ATCO came in from 2014 to 2015, they built a power line that paralleled the highway that's seen cutting north-south throughout the research site. So in the spring of 2015, so this occurred before I was out here in Alberta, our very industrious master students who are involved in this project, Cassie James and Faiza Najafi, they established a randomized complete block design experiment at 4 different sites within our Mattheis Research Ranch, which are shown here with the dots. So 2 of the sites shown by the green dots were established on soils that were more loam textured. And these soil sites were dominated by needle and thread grass, June grass, and blue grama communities, or by western wheatgrass and blue grama communities. And then we also had 2 sites established on more sandy loams or sandy dominated soils that had needle and thread grass, carrick species, and sand grasses. So this was a really, really huge project, a huge undertaking, and I was very impressed with, again, Cassie and Faiza for going and setting this up. So at each site, there were 11 treatments established with 4 replicates, and this resulted in a total of 176 plots between the 4 sites. So you can see from the number of plots incorporated in this study, not only did we have great statistical power, but this was obviously a really huge undertaking, particularly for Cassia, who was primarily responsible for planning and undertaking vegetation and soil sampling in the field. So within all of our plots, we were looking at 2 main factors of soil and plant disturbance. So we either had plots that had access mats placed and then a heavy loader driven over these access mats for a total of 16 passes, 8 at the beginning of the duration of the time that the access mat was placed for, and 8 again at the end. And then we also had similar plots where we had traffic applied at the beginning or end of the same period that matched the period of the access mat placement, except for there weren't any access mats. So we had access mats placed with traffic on top, or else traffic applied directly onto the prairie. However, we also wanted to look at how the timing of application as well as the duration of access mat application influenced our plant and soil characteristics. So access mats were placed either in the spring around April 30th for either 6, 12, or 24 weeks, or placed in July for either 6 or 12 weeks. So just so that everyone's kind of clear on this, when an access mat was placed down, there was 8 passes of our heavy loader. Then at the end of the period before the access mats were removed, we had another 8 passes. And our traffic treatments were very similar to this, except for they did not have access mats. So we had traffic applied at the same time as our corresponding 6-week treatment, and then traffic applied again at the end, just directly on our bare prairie. In addition to this, we also had untreated controls that had no traffic or no access mat applied. So we had 10 disturbed treatments and then 1 kind of control natural vegetation check to compare this to. So to give all of our treatments equal regrowth opportunities, Cassia and her team of rangeland ecologists sampled vegetation cover and biomass 1 year after treatment application in the 2016 growing season. So sampling biomass and plant cover alone took over 400 hours between 4 people over 10 years, or 10 days. Years would be kind of crazy. So this is obviously a huge undertaking and a lot of work. So the top left-hand picture is a view of the quadrant that was used to assess cover and collect biomass. And then the 2 pictures where you can see the brown paper bags, one in the middle all nicely stacked before going out to the field, and then the second to the right, which was after biomass collection. Those bags represent about half of the biomass collected, so that was 2 sites worth of biomass. So you can understand that this was quite a lot of data collection and a lot of work. In addition to this plant data, we also sampled for things like soil bulk density, we did penetration resistance, soil moisture content, infiltration rates, and some analysis of our soils of carbon and nitrogen. I won't be presenting that today due to time restraints, but I just kind of want you to understand it was a ton of work and a huge data set that we have now. So we collected plant biomass by clipping the biomass within a quadrant to 2.5 centimeters from the soil surface in order to represent approximately the amount of cow might graze. And this biomass was separated into functional groups. So we had grass and grass-like species, and then our sedges and forbs were separated into a native group that combines the desirable perennial native species, as well as an introduced group that included rudderals, annuals, early colonizers, and weedy and exotic species. And then species cover was conducted by visual counts of aerial foliar cover, and this was done for the 5 key grass species that make up the different communities within our mixed grass prairie. And again, this is the bluegrass, sandgrass, needle and threadgrass, dune grass, and western wheatgrass. So I'm going to attempt now to take you through some of the results of Cassia's plant analysis. Please keep in mind, again, I'm not a plant scientist so much as a soil microbial ecologist, but I'm going to try to do justice to the huge data sets that she has. So looking at some results here, this is the grass biomass in grams per meter squared as affected by our direct traffic. So again, traffic was applied at the beginning of the period and at the end of the period. So for example, our early 6-week plots had traffic applied in the spring and then again 6 weeks later, versus our late 6-week plots had traffic applied in July and then also 6 weeks later. So when we looked at our grass biomass, we found that grasses and grass-like vegetation were found to be the bulk of our vegetation. So we had about 66% on loam and 74% on sand. So when we looked at the impacts of direct traffic on our grass biomass, we found that there were no significant differences in grass biomass on either soil texture sites. However, when we compared the mitigative effects of using access mats, we did find some significant results with respect to grass biomass, which was soil texture specific. So shown here is again our grass biomass, specifically within our loam textured soils. And this is showing our paired comparisons of access mat with traffic applied versus traffic directly onto the prairie. So for our loam textured soils, we found that there were no significant difference between these treatment pairs. However, when we look again at the same comparison of grass biomass between the access mat plus traffic or traffic only plots within our sandy sites, we found that both long and late placement of access mats resulted in a significant reduction in grass biomass under the mats between these 2 treatment pairs. So if access mats were applied early in the spring for only 6 weeks, there was no significant difference in our grass biomass on the sandy sites. However, applying access mats for longer than 6 weeks or later in the season resulted in a significant decrease in our grass biomass when access mats were applied. So this indicated that applying access mats to prairie after the vegetation has started to actively grow had negative impacts on our native grass biomass. However, again, these impacts were only seen on sandy textured soils. So we've looked at how our grass biomass responded, and we're now going to switch over to our native Forb biomass. So again, this is our native Forb biomass in grams per meter squared, and we're looking at the effects of direct traffic on our native forbs. So native forbs made up approximately 21% of the vegetation on loamy sites and 16% on sandy sites. And for native forbs, when direct traffic was applied twice, so at the beginning and at the end of the period, from 6 to 12 weeks, this resulted in a significant reduction in biomass, and only the plots that had a 24-week separation between the traffic applications did not have a reduction in biomass. However, placing access mats early in the season for 12 weeks resulted in an increase in native Forb biomass for both soil textures. So this is just showing our loamy soils right now, but this is true for both soil textures. So when access mats were applied early in the spring for 12 weeks, we had a significant increase in our native Forb biomass. And this is due to the fact that native Forb regrowth is likely from the seed bank and reflects reduced competition with the grass biomass at this point. We found, changing again gears, to introduced Forb biomass, we found that there was no significant impact of direct traffic on our introduced Forb biomass for either soil texture, regardless of time between traffic passes. However, again, applying access mats influenced introduced Forb biomass differently between the sandy and loamy soils. So in loamy soils, which are shown here, access mats placed in the spring for either 12 or 24 weeks resulted in a significant increase in introduced Forb biomass. Whereas in sandy soils, access mat placement and duration at any time except for that early 6-week placement in the spring resulted in a significant increase in introduced Forb biomass. Finally, I'm just going to present one last graph here of Cassia's results looking at the change in grass cover in a percentage by species. And this is a comparison between our access mat with traffic applied plots and our traffic only treatments. So starting on the left with our first species, our blue grama, we found that short and early placement increased cover while all other treatments decreased cover. For our sandgrass, we found that early and long placement significantly decreased the cover, and again, this was a sandgrass, so it was only found on our sandy sites. For our next species, needle and threadgrass, we found that all access mat placements resulted in significant decrease in cover. For our for our June grass, our short and late placement did not significantly decrease cover, but all of the others did. And then for our western wheatgrass, only the later 12-week treatment resulted in a change, which you can see here was an increase in cover. So this was a lot of data to throw your way, which is just a small fraction of our overall data for this project so far, but what does this all mean? Well, we found overall from just looking at our analysis of the plant community data that grass biomass was negatively impacted by access mats on sandy soils, while loamy soils had no significant differences. We found that native Forb biomass was reduced when traffic was repeated twice within 12 weeks, and increased when mats were placed early and for long durations. We also found that introduced Forb biomass increased when mats were placed early and for durations of 12 weeks or longer. And finally, we found that in general, grass cover decreased with access mat placement over 12 weeks. So our general conclusions on access mat use from this plant data is that sandy soil textures are more sensitive to the impact of access mats, and this impacted grass and introduced Forb biomass. We also can conclude that longer durations of axis mat placement increased or introduced Forb biomass, and that axis mat placement early in the spring and for longer than 6 weeks changed grass species cover overall. So this would lead us to conclude that axis mat should be applied in the spring for 6 weeks or less in order to decrease any negative impacts on mixed grass prairie biomass and plant cover. So we also obviously didn't want to just look at plant characteristics, but also at soil characteristics. So I'm going to take you through now some results from our soil and soil microbial analysis. Again, because we're trying to get an overall idea of how the ecosystem is changing and functioning in relation to these types of disturbances. So I hope I can illustrate to you today how important the soil microbial communities are as a microbial ecologist. So the microorganisms that live in soil are essential moderators of the ecosystem goods and services that are provided by soils. So in the past, soil microbial communities were often kind of thought of as this black box where we knew they were involved in different processes within the soil but didn't really know their specific role or how important they were. However, now we've had an advent of different techniques over the last 20 or so years, so we know a lot more about their roles in soil. So we know that they're integral moderators of the formation of soil structure, in mediating carbon, nitrogen, phosphorus, and sulfur cycling, and in the promotion of plant growth, to name a few. And the major functions that a soil provides, again, things like regulating water quality, mediating greenhouse gas fluxes, promoting plant growth, mediating nutrient cycles, these are all coupled tightly with the dynamics of the soil microbial community. And this is really important to keep in mind when we're trying to think of the effects of microbial community changes with respect to soil health and overall ecosystem functioning. So soil health is defined as the continued capacity of a soil to function as a vital living system in order to sustain biological productivity, promote air and water quality, as well as maintain plant and animal and human health. So in assessing soil health, we can get a much better idea about the overall functioning and sustainability of that ecosystem. So we can measure soil health through biological measures such as the abundance and diversity of soil microbial communities. And microbial measures of soil health often respond quickly to changes in agricultural management or to different disturbances, which makes them really good indicators for short-term changes in soil health and overall ecosystem functioning. So assessment of soil health must be included within the development of best management practices for improved ecosystem and environmental sustainability. And this is because, again, a healthy soil will equal healthy grasslands that are more productive and can contribute more with their goods and services. So studying soil microbial communities is not an especially easy feat, and this is because of the huge biodiversity that soils can support. So to put this into context, there are the same number of stars in our galaxy as number of bacteria in a kilogram of soil. And if we went out into the prairies and took up some nice AH horizon from a nice ternism, within a teaspoon alone, the number of different microbial species, so not cells, but different species, can be within the millions. What complicates the study of these microbial communities further is that the majority of these soil microbial communities are what we call viable but not culturable. So this means that although they are living in the soil, they're active, they're involved in different processes, they cannot easily, if at all, be grown in a lab with traditional microbial methods. And this relates back to this huge biodiversity that the soil supports. So to circumvent this, soil microbial ecologists such as myself now heavily rely on molecular methods to study soil microbial communities. So this helps us circumvent what's called the Great Plate Anomaly, where basically when we try and grow and plate microbial communities, we are often only getting between 1% to 5% of what's actually in the soil. So instead, we use molecular biology, and this relies on the central dogma of molecular biology, which tells us the expected direction of genetic flow within a cell. So DNA codes for genes, which if upregulated will be translated to RNA and can then be result in protein synthesis. So within a lot of the work I do, I look at DNA and RNA. Within this project, we're only looking at DNA. So we can extract DNA from soils. We can then target specific either taxonomic or functional genes in order to look at either species identification or overall functioning. So we can look at genes that code for specific enzymes involved in different reaction pathways in the soil. And we can use different methods such as qPCR or quantitative polymerase chain reaction in order to enumerate these genes to get an idea about the size of these functional communities. We can also use next generation sequencing platforms such as Loamy to look at the overall diversity of our communities. And then by pairing molecular methods with stable isotope approaches, we can also do things like track carbon use through the community. So overall, with these methods, we can do things like relate changes in the abundance and diversity of the soil microbial communities to changes in ecosystem processes, which they are in fact driving. So my kind of main interest in soil microbial communities is due to their role in producing and consuming greenhouse gases. So in Canada, agriculture accounts for approximately 10% of annual emissions. And in Alberta, agriculture and forestry accounts for approximately 9% of annual greenhouse gas emissions. So although this number sounds small, this is equivalent to about 25 megatons of emissions annually. And with Alberta's new carbon tax being phased in, that small amount is actually equal to approximately $750 million a year in carbon taxes that will need to be paid out by land managers. So this means that there is a huge potential to influence the microbial communities responsible for these emissions through our land management and proper mitigation of large disturbances in order to enhance soil carbon storage as well as decrease emissions and hopefully decrease the future carbon taxes for producers and land managers. However, we'll only be able to make recommendations for management and practices to decrease these emissions if we better understand these soil microbial communities and what influences them. So of particular interest to me is are the microbial communities involved in nitrogen cycling. So shown here is the terrestrial nitrogen cycle. And I'm interested in nitrous oxide emissions because nitrous oxide is a potent greenhouse gas with a global warming potential about 296 times that of carbon dioxide. So globally, soils account for approximately 36% of annual nitrous oxide emissions. And the two major groups that produce and consume nitrous oxide from soils are the nitrifying bacteria and archaea and the denitrifying bacteria. So the abilities to denitrify and nitrify are common to many different species of microorganisms, which makes the use of molecular targets such as DNA ideal for their study. So because these microbial communities are responsible for this production and consumption of nitrous oxide from soils, how they change in response to things like disturbance and how these changes relate to nitrous oxide production may allow the creation of predictive models for use in the development of best management practices to decrease nitrogen losses and hopefully those carbon taxes. So in order to study these bacteria and archaea, I basically target the genes that code for enzymes that are involved in the nitrification and denitrification reaction pathways. So to study these microbial communities, we sampled soil from 2 of the 4 grassland sites that Cassie and Mattheis set up in 2015, so 1 sandy and 1 loam soil. And we had to choose a subset of the plots. Unfortunately, due to the costly nature of molecular biology, we couldn't do all 11 treatments at each site. So instead, we focused on using our natural vegetation check, as well as our direct traffic and axis mat with traffic plots that had axis mat and traffic applied in the spring and then again after 12 weeks. So I extracted DNA from soils, which is very simplified here, but it's a series of binding, washing, and elution steps basically. I then used quantitative polymerase chain reaction in order to quantify the number of genes present for different nitrogen processes in the soil, as well as taxonomic gene targets. So this led us to basically look at the size of different communities involved in these processes. And then currently we're also creating amplicon libraries in order to use next generation sequencing for diversity analysis. So I'm not going to be talking about that more today, as that's still ongoing. So I'm going to take you through just a couple of really basic results of some of our soil physical and chemical characteristics. So shown here is our soil moisture on a gravimetric basis in 2015 and 2016 at the time of sampling in our control, axis map plus traffic plots, and then traffic only plots. So overall, we found that our loamy soils had higher moisture than sandy soils, also had higher soil nitrogen and carbon. However, we found that overall, axis mats had increased soil moisture in year 1, so in 2015, than what was seen in the control plots. However, by our second year in 2016, so a year after those treatments were applied, direct traffic showed a positive effect on soil moisture. There was an increase in soil moisture in the plots that had traffic directly applied on the prairie. And this also paralleled an increase in soil compaction, bulk density, and slowed water infiltration rates in 2016 in our direct traffic plots. So although we didn't see much impact of our direct traffic on our plant data, we did see differences when we looked at our soil physical properties. So shown here are how the different disturbance treatments influenced kind of what you can think of as soil chemistry. So on our left we have pH, then we have soil nitrogen and soil carbon. Again, in our control, AM, which is axis mat plus traffic, and then our direct traffic, which is denoted by TON plots. So we found that direct traffic had higher pH than our axis mat plots. And we also found that direct traffic applied onto the prairie increased soil nitrogen and carbon compared to both our control and axis mat plots. So we saw that although there were not distinct effects as much with our direct traffic on our plant community data, that we did see definite influence of the direct traffic on both our soil physical and chemical characteristics. So I'm going to just share a few results from the microbial analysis before I wrap up. So I'm going to try and keep this simple. Shown here is what we call our 16S ribosomal RNA gene copy, and this is on a per gram dry soil basis. So this gene is basically common to all bacteria. So you can think of it very loosely as a proxy for the total bacterial community size. So we found that when we looked at the size of our total bacterial community, despite those changes in plant composition in some instances, as well as in biomass and changes in physical and chemical soil characteristics, that the overall bacterial community size was not different between our different traffic and axis mat with traffic treatments. We did find that there was a significantly larger bacterial community size in our loamy versus sandy soil, but not between our treatments. So this is part of why we're now going to be running sequencing on this to look at the diversity, because although the overall size of the communities may not change that much between the treatments, there may be a distinct change in diversity. So we'll see. We also found that some of our denitrifying gene targets that were involved in nitrous oxide production followed similar trends. However, there were definite effects of our different traffic treatments on some of our other gene targets. So shown here is our 18S ribosomal RNA gene copy per gram dry soil, and this is a proxy for the total fungal community size. So we found that there was differences in how these treatments influenced the size of the fungal community between our years. So in 2015, when it was close to when our traffic treatments had been applied, we found that both types of disturbance, whether it be axis mat plus traffic or direct traffic, decreased the size of the fungal community compared to what we saw within our control plots in 2015. However, by 2016, so a year later, again, when we saw all those impacts still were existing from our direct traffic on our soil physical and chemical characteristics, the direct traffic plots had a positive effect on the fungal community and had a larger fungal community size. We also saw some differences for our nitrogen cycling genes or community sizes. So shown here are our log ammonium monooxygenase gene copies per gram dry soil. So this is a proxy of the size of the nitrifying community that's involved in nitrous oxide production. We found that the two traffic treatments influence the size of the nitrifying community differently compared to the size found in the control over both years. So in axis met plots, we had a higher or larger nitrifying community, and in the direct traffic plots, we had a smaller nitrifying community. Additionally, I found that some of the nitrogen cycling communities, such as our NOS or nitrous oxide reducing community, were also affected. So our NOS gene copies per gram dry soil represents our nitrous oxide reductase gene. So this represents the reduction or consumption of N2O, so our greenhouse gas, to dinitrogen gas. So basically, a larger population would indicate a potential higher for nitrous oxide consumption, which is a positive thing from a sustainability viewpoint. So we found that the different traffic applications affected the size of this community differently depending on the texture of our soil. So we found that axis mat use decreased the size of the nitrous oxide consuming community within the sandy soil, while direct traffic increased the size of the nitrous oxide community. However, in our loamy soil, both types of traffic treatments decrease the size of the nitrous oxide consuming community in the loamy soil. So what does this all mean? Well, we found that direct traffic influenced both soil physical and chemical characteristics, and we still have huge data sets of soil data to go through and work through. When we looked at our microbial analysis, we found that the size of the total bacterial community and some of our denitrifying communities that are involved in nitrous oxide production were unaffected by traffic treatments, but differed between soil textures. So again, we're continuing to look at the diversity of these communities as well. However, we did also find that the size of the fungal community, the nitrifying community, as well as the N2O consuming community were all influenced by the use of axis mats and direct traffic, and some of these effects were soil texture specific. So again, we have a ton of data, and since we want to look at the overall ecosystem functioning, we need to now try and integrate our soil, plant, and microbial data. And again, we need to continue with our sequencing work to be able to assess microbial diversity as well within these plots. Additionally, I'm also pretty excited about this. I'm just going to mention pretty quickly. But I'm also currently in the planning stages for a lab incubation where I'm going to assess how carbon use patterns change after axis mat use. So again, we saw from Kathy's data that there was change in biomass levels between her functional groups based on axis mat use, as well as a change in her plant species or cover composition. And because of that, I want to assess how or what carbon sources, whether it be residues from the plants or from our soil organic matter, do soil microbial communities utilize after this axis mat removal, so after we've had this kind of plant die-off and change in composition, and how changes in this carbon use relate to carbon dioxide production. So I'm going to be using 13 carbon labeled plant residues in a soil incubation and using both molecular methods as well as 13 carbon phospholipid fatty acid analysis and delta CO2 analysis to track carbon use through the community. So we still have a lot of data to go through and a lot of work to do, but we've had a lot of really interesting results so far, and I think that this project has been really important with respect to creating recommendations on axis mat use in our prairies and to ensure that these valuable ecosystems remain healthy and productive. So overall, we found that sandy soil textures are more sensitive to the impacts of axis mats, that longer durations of axis mat placement increased introduced Forb biomass, that axis mat placement early in the spring and for longer than 6 weeks changed grass species cover overall, and that some nitrogen cycling microbial groups as well as our fungal communities responded to the types of traffic disturbance differently, suggesting different disturbances may alter the potential of these microbial communities to produce and consume nitrous oxide and therefore influence associated ecosystem functioning during recovery. So for rangeland managers, the implications of these results are that long duration and late placement of axis mats may increase negative impacts on plant biomass and community composition. That lawn axis mat placements require longer vegetation recovery times, and placement on actively growing vegetation may impact the vegetative recovery and resilience. Also, the use of access mats in Alberta mixed grass prairie may need to be determined based on soil texture in order to minimize their impacts on soil microbial communities. And finally, different industrial disturbances may impact soil microbial communities responsible for ecosystem processes such as the production and consumption of nitrous oxide, which should be considered in establishing proper best management practices in order to promote ecosystem sustainability and reduced greenhouse gas emissions. So in conclusion, again, we're still working through this and we'll see what our other data says. But at this point, it looks like for mixed grass recovery, early and short duration of access mat placement is recommended in order to limit negative impacts to plants, as seen from longer duration and late season access mat use. And additionally, I think that it's important to point out that soil texture should really be determined before access mat placement in order to assess whether these access mats may positively or negatively impact microbial communities and therefore ecosystem functioning. So just lastly, I want to thank all the people who have helped in the field and in the lab, as well as our funding sources. And I'm happy to take any questions.
If anyone in our audience has any questions, please feel free to type it in the question section of the webinar dashboard. Oh, there's a question here by a listener named Dale. Were the impacts of your direct traffic treatment 8 passes with a loader comparable to the actual direct traffic amounts and types incurred during power line construction?
Good question. So in total, there was actually 16 passes over the period, 8 at the beginning and 8 at the end, which is probably a fairly low amount of passes for a construction site. So regardless of that, there was still an impact of the direct traffic on some of our other soil properties. So in general, I would probably think that there would be a larger impact potentially on our soil or on our plant communities as well, if under more intense direct traffic passes.
Thank you. There's another question from a listener named Sarah. Were traffic treatments ever applied during wet or rainy conditions?
So the access mats, first of all, were kind of proposed in order to travel on the saturated soils through the boreal forest. When they were first applied in the spring of 2015, there was a delay in spring emergence, and I believe that was because it was a fairly wet spring, but I'm honestly not sure in the spring how wet it was compared to normal Albertan springs, being a new Albertan, but in July it would have been very dry.
Thank you. We don't have any questions at the moment, but if there are any listeners out there who would like to ask a question now, please feel free to type it in. I would just like to mention that this webinar has been recorded and it will be uploaded later today to the PCAP YouTube channel, and that's youtube.com/user/skpcap, and you're welcome to pass that link on to anyone who is not able to make it today. I also like to encourage everyone in the audience to take a look at the PCAP website and check out the other events that we have going on for Prairie's Got the Goods Week. Just click on Events and Ecological Goods and Services Week 2017 for the program that we have there. All of the events are free and you just have to register for the webinars just like you did for this one here. I guess if there aren't any more questions, I would like to say thank you everyone for coming, and thank you very, very much, Karen, for this amazing presentation. I know I learned a lot and I got a lot out of it. If anyone has any questions for Karen, please feel free to send her an email, and her email will be sent to you after this webinar. You will receive a quick survey when you're done this webinar. It'll just take a minute. If you don't mind filling that out, we would really appreciate it. So thank you very much, Karen, and thank you to all of our listeners today.
Thank you.
Have a great rest of your day.